After talking about the process of human development, it makes sense to step back a little bit and talk about stem cells. We know that each of us started out as a single fertilized egg, a zygote, and we have developed into whole people. We created different cell types, different tissues, different organs from that single cell. How did that happen? Well, first, I want to start by defining what a stem cell is and discussing what a stem cell is not. Stem cells, by definition, divide differently than non-stem cells. So, for example, we see the stem cell, and when it undergoes its first round of cellular division, it creates two different cell types. One, another stem cell, and two, a stem cell or a cell that is destined to be differentiated into some other type of cell, in this case, a red blood cell. The stem cell can go on to subsequently divide again to create another stem cell and another cell that will be destined to differentiate. Stem cells then can divide indefinitely, meaning they are immortal. What stem cells are not are necessarily cells that can become any type of cell. That word, the word that we use to describe that, is potency. There are different levels of potency. If a stem cell has the capacity to develop into any type of cell, we describe that stem cell as a totipotent stem cell. It can become anything. Totipotent stem cells are only present in fertilized eggs until the end of the morula stage. After the cell enters the uterus, it becomes a blastocyst. The cells in the inner cell mass of that blastocyst are described as pluripotent, meaning that they can become almost anything, but not everything. Remember that on the outside of that blastocyst, we have the trophoblasts that will become part of the placenta. So the inner cell mass, the cells of the inner cell mass cannot become the placenta. Therefore, they are not totipotent, but rather are pluripotent. In adults, we have two different types of stem cells, those that are described as multipotent. For example, the stem cells that are located in our bone marrow that give rise to all of our different blood types, or they can be unipotent. For example, the cells that are in the bottom layer of our skin that give rise to new skin cells when we slough off skin cells, new gastrointestinal cells when we slough off gastrointestinal cells, new lung epithelial cells when we slough those off. Those stem cells that reside in those specific organs can only become cells of that type. So they are described as unipotent. We can acquire stem cells from a number of different sources, from the embryo, from the umbilical cord, or from adult tissue, although cells acquired from adult tissue have limited capacity or they must be treated in specialized ways. In the embryo, if we have any cells derived from something before the blastocyst, specifically the morula, those cells are totipotent. Cells in the blastocyst, in the late blastocyst, or beyond that are pluripotent because they cannot become the placenta. And in fact, the longer we go in human development, the more specialized cells become, and therefore the less potent they become. It's possible to derive stem cells from umbilical cord blood, and the cells that we derive from umbilical cord blood can become blood cells, epithelial cells, endothelial cells, meaning the cells that create our blood vessels, or neural cells. There are some things missing from this list, so umbilical cord blood provides us with pluripotent but not totipotent stem cells. We can derive stem cells from adult bone marrow, but those cells can only give rise to other types of blood cells, so these are described as multipotent stem cells. It's possible to acquire pluripotent stem cells from adult human tissue, but it does take some manipulation. We can acquire stem cells from adult skin cells via a process called somatic cell nuclear transfer. Somatic cells are simply cells that contain a normal nucleus. In other words, any cell in our human body outside of our sperm or our egg. If we harvest an adult cell, for example, a skin cell, the next step in somatic cell nuclear transfer is removing its nucleus. We're going to take that nucleus and put it inside of a fertilized egg. So we're going to remove the nucleus from the fertilized egg and replace it with the nucleus from our somatic cell. We're going to allow that egg to develop until the blastocyst stage, and we're going to remove the cells that will become the embryo, specifically the inner cell mass. We're going to allow those inner mass cells to reproduce, and when we have a lot of cells, we're going to add chemicals. These chemicals will induce differentiation into different cell types. The different chemicals that we use, each of them are going to induce the generation of a different cell type. That is somatic cell nuclear transfer. It's also possible to induce pluripotent stem cells by isolating a somatic cell like a skin cell and adding to it both genes and chemicals. The addition of genes, DNA, and chemicals will create what we call an induced pluripotent stem cell. Here's how that happens. Again, we're going to isolate adult skin cells, and we're going to add a number of genes. The genes that need to be added to the DNA of these skin cells are genes for parts of our DNA that are going to regulate how the DNA is going to be used. Those genes therefore reprogram the skin cells to become induced pluripotent stem cells. That's the iPS. We will add chemicals to the induced pluripotent stem cells, and again, depending upon which chemical we add, we can induce those adult skin cells into a number of to differentiate into a number of different cell types. Either muscle cells, fat cells, neurons, pancreatic cells, blood cells, etc. The benefits of using stem cells in scientific research and in medical treatment include the following. If we can create new neurons, nerve cells in a culture dish, either by treating embryonic cord blood or adult stem cells that have been converted into, or adult cells that have been converted into stem cells, if we can induce the creation of nerve cells, we might be able to treat diseases like Alzheimer's disease or Parkinson's disease, where specific regions of the brain have had neurons die in them. If we can replace those neurons, we may be able to stall the progression of these two neurologic diseases. Alternatively, if we can create pancreatic cells, it's possible that we can treat or possibly cure diabetes. These pancreatic cells, remember, are responsible for producing insulin. People with type 1 diabetes are incapable of producing their own insulin. And if we give those individuals stem cells, pancreatic stem cells, it's possible that they would be able to create their own insulin. People with muscular dystrophy have muscle cells that are dying. If we could replace those dying muscle cells with muscle cells that were derived from stem cells, we could treat and possibly cure muscular dystrophy. Each of the sources described in this video provides different avenues from which to acquire them.